Bon matin tout le monde. Good morning, everybody. It is our pleasure to introduce Dr. Jessica Prodger for the basic science plenary session. Dr. Prodger is an associate professor in microbiology and immunology at Western University, where she holds a Canada Research Chair and serves in a scientific leadership role with the Reach Martin Delaney, I'm pretty sure this is not the right way to say it, Collaborative for HIV Cure Research. Our research program spans mucosal immunology and HIV pathogenesis with a focus on understanding the drivers of inflammation in the genital mucosa. Our work has advanced our understanding of penile HIV susceptibility and neovaginal health in trans feminine individuals, while more recently concentrating on HIV persistence during antiviral therapy. In particular, she co-lead a longitudinal HIV reservoir cohort in Uganda, where she is also a long-standing research collaborator working to define the host and viral mechanism that shapes the replication-competent HIV reservoir. Today, she will share insight into how longitudinal sampling can strengthen causal interference in HIV persistent research and help proving biologically meaningful drivers on reservoir maintenance. Please join us to welcome Dr. Jessica Prodger. The clicker is there, and you have water just
It's not somebody else's water.
That's okay. A little immunity is good. Good morning, everyone. Thank you so much for the introduction. And thanks for the opportunity to speak to you this morning and getting up so early. It's really an honor to be giving this talk here at CAHR, and it is just fabulous to be back in Winnipeg. So I'm just going to see, does this laser actually point? No. Okay. All right, these are my conflicts. So I want to start with a really broad overview of why HIV persists on antiretroviral therapy, and a big picture view of the types of cures that are under investigation. So this is meant to be a CD4 T cell that's activated. It's fighting some infection in the body. And activated T cells make a lot of proteins, and they're also much easier for HIV to infect. When HIV enters the cell, it has to integrate its own genetic material into that host cell's DNA, and then the host cell will make the HIV proteins inadvertently along with its own. And these assemble into viruses, they bud off of the cell, and this is how the infection spreads. Now, the vast majority of the time, these cells die, either through apoptotic effects, they realize that they're infected and they kill themselves, or those HIV proteins are recognized by other immune cells, cytotoxic immune cells in the body, and they clear the infected cells. However, in a minority of cases, these cells can revert to a resting state, and they stop making HIV proteins, so they're effectively invisible to the immune system. And the progeny of these cells can persist for many decades. But if they become activated again, they can start making HIV proteins and virus. And this is why we need lifelong antiretroviral therapy. And it's this population of clonally maintained and long-lived cells that has integrated HIV DNA or provirus that we need to deal with if we want people to be able to stop taking antiretroviral therapy. So how are we trying to do that? One approach is to just prevent these cells from ever becoming reactivated again, permanently silence them, and they can live happily in the body without causing any problems. An alternative approach is to drive them to reactivate and push them down that pathway in the setting of ART so that the virus that's made can't go on to infect new cells, but then to enhance the immune control, so enhance apoptotic pathways or enhance the killer cells of the immune system to clear them. And yet another approach that's being investigated is gene editing. And this can be targeted towards the provirus itself to make it inactive, or it can be targeting uninfected cells to protect them from becoming infected. And I think everyone agrees that it's not going to be any one of these modalities in isolation. We're probably going to need a combination or all of them to be able to reach a functional cure. So one complication to all of this is that actually the vast majority of provirus in cells is defective and it can't go on to make infectious viral particles. So in collaboration with Gwyn Lee through the REACH Martin Delaney Collaboratory, we performed near full-length sequencing on proviral sequences from 23 people from Rakai, Uganda. And the horizontal lines that are here on the screen are showing you the proviral sequences from one person. And those ones at the top, the blue ones, those are the intact ones that could actually go on to make infectious virus. And everything else below that has major mutations or deletions in it, so it's defective. And when Gwyn looked at all 23 participants in the 607 proviral sequences that she made, she found that only 5% of these proviruses were actually intact. And if our goal is that a person can stop taking ART, it's these intact proviruses that we need to focus on. Unfortunately, it's tricky to measure just the intact ones, but it's really important to, because there's accumulating evidence that cure modalities act differently on intact or defective proviruses, especially those that are trying to drive reactivation and increase those apoptotic or killer cell pathways and that rely on HIV proteins. And so one of the ways that we can try to measure intact proviruses only is by expanding them ex vivo and seeing that they're replication competent. And one of the assays, one of the sort of earliest assays we had for quantifying the intact proviral reservoir was a quantitative viral outgrowth assay. So in this approach, cells are isolated from a person, and then just those resting, long-lived cells are then isolated further. And these resting cells are plated out at limiting dilution. And then they're given a really strong stimulus to try to reactivate the latent provirus. And then that provirus can replicate within that well, amplifying the signal, and this can be measured using an ELISA. However, the problem is that this is an underestimation of the reservoir. So not all proviruses are reactivated in this one single round of reactivation. And Yachi Ho, I think when she was working in Bob Silicano's lab, she showed this really beautifully. So she did full genome sequencing on a number of people and also QVOA outgrowth. And she's representing each individual person here with these half-circle, gobstopper-looking things. The big purple circle is the total number of cells that contains any HIV provirus. The pink circle is the proportion of those proviruses that's intact. And that little yellow circle in the middle is the proportion that grew out with the QVOA. And so what you can immediately see is that the QVOA underestimates the intact reservoir. But also the degree of underestimation is differing between people, making the QVOA an imperfect surrogate of intact reservoir size. But I think despite this imperfection, it might actually be measuring something different that might be equally important, because some people appear to have a reservoir that is much easier to reactivate than other people. So if you look at person number 16 that's on the top left, you can see that yellow circle fills up a lot more of the pink than someone like participant 20. So an alternative strategy to try to measure intact proviruses is to look at the genomes. However, performing full genome sequencing at a single genome level is very labor intensive, and it's completely inappropriate at a cohort level. But it turns out that if you look at just two short regions in the genome, so here represented by Psi and OV, if those two regions are there, there's actually a really good chance that the whole genome is intact and is there. And so the intact proviral DNA assay, or the IPDA, relies on droplet digital PCR technology. So each droplet where the PCR reaction is happening, and each droplet contains at most one single genome. And then the readout for this is like flow cytometry. So you can see on this plot here on the y-axis is a Psi signal, and on the x-axis is OV. And if a provirus is defective for ONV but has psi, it'll show up there where those blue dots are, the reverse for the green dots, and this small population of orange dots, I don't even know if you can see it, but that's in the top right-hand quadrant. Those are the proviruses that have both and are presumed to be intact. But a limitation of this assay is it's very sensitive to genomic diversity. And it was designed based on subtype B HIV, which is the predominant subtype circulating in North America, Europe, and Australia. And when we applied this assay to cells from our cohort, which is in Uganda, where subtypes A and D are dominant, we found that it would often, very frequently fail in one of these two regions. So a number of people, including ourselves, are working very hard on adapting this assay for use in global cohorts. If you want to hear more about that, you can come to the Basic Sciences oral abstract session this afternoon. Sarah Gownlock is going to be talking about applications in Uganda, and Maggie Duncan about how subtype diversity in subtype B, or genetic diversity in subtype B, affects this assay. Okay, so as promised, talking about lessons learned from an observational cohort in Uganda. So the Rakai latent viral reservoir cohort is located at the Rakai Health Sciences Program, which is located in Rakai district in Uganda. And Uganda is a small country in East Africa, and the Rakai district, if you look on that middle map, it's on the border with Tanzania and also borders with Lake Victoria. And not only was one of the hotspot regions for the HIV epidemic in Africa, in southern Uganda, but additionally, the Rakai district has really epidemiologically distinct populations within it. So we have bustling border trading centers along the border with Tanzania, where the Trans-African Highway enters and crosses the country. There's also very remote but very high-risk fishing communities along the shores of Lake Victoria and on the islands. But we also have sustenance agricultural rural communities more in the central region. And the Rakai Health Sciences Program is also home to an NIH International Center for Excellence in Research, so it has quite a well-equipped lab with very well-trained staff. So why Uganda? Well, we know that the vast majority of people who are living with HIV are living in Eastern and Southern Africa. And you can see on that map, Uganda there just to the right of the DRC, has quite a high HIV prevalence. And most HIV cure research is being performed in North America and Western Europe and Australia due to global wealth distribution inequities. And there's substantial differences between the HIV epidemic and people who are living with HIV in these two regions. And two of those differences that I'm going to talk about today are sex distribution. So you can see that the majority of people represented in purple here living in Africa with HIV are female, as opposed to males in North America. Although I learned yesterday at the opening session that this sex type distribution may be especially important to the Manitoba epidemic. And also global subtype distribution. So whereas subtype B dominate in North America and Europe, in Africa it is subtypes C and A followed by D. So to increase African representation in HIV cure research, Tom Quinn and I, who at the time was at the intramural program in the NIH at the National Institute of Allergy and Infectious Diseases, we established the latent viral reservoir cohort at the RHSP. And we established this in 2015, enrolling 70 participants. And in collaboration with Bob and Janet Silcano, we performed the QVOA assay on all 70 participants. And this represented the first description of the latent viral cohort, the latent viral reservoir in an African cohort. So we've now followed this cohort for 9 years, with the 10th year of follow-up underway. And Tom Quinn is now retired from the NIH, so I have the pleasure of working with Andy Redd and Steve Reynolds, and also my dear friend Ronald Galiwango at the Rakai Health Sciences Program. And by following this diverse group of ART-suppressed individuals over nearly a decade, we've started to see that reservoir persistence is actually a dynamic process that's shaped by host biology, viral genetics, and treatment exposure. And so I'm going to share with you 3 observations that we've learned about HIV persistence through this cohort. And the first is biological sex and HIV persistence. So we often think about biological sex, especially in the basic sciences, as being binary and also static, determined at the moment of conception based on whatever chromosome complement is delivered. But biological sex is neither of these things. Many people, including both people assigned male or female at birth, have an extra copy of the X chromosome that carries loads of immune genes. And things like epigenetic modifications and sex hormones also dictate sexual characteristics. And these can be very dynamic, changing over the lifespan and influenced by exposures. For example, gender-affirming medical care. So the need to consider gender when thinking about biological sex. So when we're asking about whether or not biological sex influences the reservoir, we need to think beyond a simple binary and consider the different biological layers that may shape HIV persistence. And there's a lot of evidence out there that biological sex is really important to HIV. Back before the days of test and treat, we could observe that females early in infection had much lower viral loads than males, but they progressed more rapidly to AIDS. The baby cure study in KwaZulu-Natal in South Africa found that female infants were much more likely than male infants to be born with HIV through vertical transmission. And we know that females are more likely to spontaneously control HIV. They're more likely to be elite controllers with undetectable viral loads in the absence of ART. Now, unfortunately, females have remained really underrepresented in HIV cure research. And I think we got away with this in the design of antiretrovirals because antiretrovirals are targeting the virus. But I'm not sure we're going to get away with it in cure research because many cure modalities are targeting host factors, things like immune pathways, for example, TLR7, which is located on the X chromosome, or epigenetic and host genes. And these attributes may differ by sex in meaningful ways. So the Rakai latent viral reservoir cohort is particularly well suited to look at biological sex. It's a generalized epidemic in Uganda. The dominant mode of transmission is penile vaginal sex. And so we have good representation of both males and females, and they're well matched for covariates like access to HIV care, age, co-infection, exposure, et cetera. So when we went to look at our QVOA data in the reservoir cohort, we actually found that females had significantly less viral outgrowth compared to males. However, when we then went to look at total HIV DNA, we found there was no difference between the two sexes. And so using a page out of Yachi Ho's book, although my circles are not 3D, what this graph is showing is that males and females have the same amount of total HIV provirus, that's the blue circles, but the females have less QVOA outgrowth. And so what's still missing from this picture, of course, is the amount of intact proviruses, which hopefully we'll get soon from the IPDA data. But I think we're left with 2 possibilities to explain this data. One is that females truly do have a smaller replication-competent reservoir, and that's why we're seeing less outgrowth. But the alternative explanation is that it's just poor latency reversal. We're just not able to reactivate their cells as easily. And there's a fair amount of data in the literature to support this latter hypothesis. For one thing, estrogen has been shown when binding to the estrogen receptor alpha to translocate to the nucleus and bind the HIV promoter, the LTR, and suppress HIV transcription. A genome-wide screen has identified the estrogen receptor alpha as a critical determinant of latency. And this has also been shown in vitro. So this graph is showing reactivation of a cell line with latent provirus. And you can see at resting there's very little reactivation. But when a latency reversal agent, CAHR or vorinostat, which is an HDAC inhibitor, is included, you see more reactivation, as expected. But when tamoxifen is included in this, which blocks signaling at the estrogen receptor, we see even more reactivation. And when estrogen is augmented in the model, the effect is attenuated. So fast forward a few years later, and we now have several years of longitudinal QVOA data on the cohort. And so we hypothesized, okay, if estrogen is suppressing reactivation in the QVOA, we should see that repression released when women in our cohort go through menopause and estrogen levels drop. But in this real-world observational cohort, as in other observational cohorts looking at longitudinal QVOA data, you see there's a huge amount of variability in the data. So we teamed up with Art Poon and his postdoc, Rusil Freire, and she took a Bayesian hierarchical approach, and she modeled the QVOA data at an individual level. So this allows each participant to have their own slope, while also borrowing strength from individuals with incomplete data. So even people who only have a single observation can contribute information to the population level estimates. And this model has really contributed to the power of the cohort because it's allowed us to draw a lot more stable conclusions from this sort of noisy longitudinal data. And we found when we looked among women in our cohort that transitioning through menopause was associated with a 1.8-fold increase in outgrowth. So in summary, biological sex is not binary or static, and sex hormones can affect both latency reversal and the assays we're using to try to quantify the reservoir. And as a result of this, I believe we really need better representation of women in HIV cure research, and we need to be considering biological sex in a much more granular and intentional way. So the second thing that I want to move on to now is how the virus can influence HIV persistence. So one of the barriers to reducing the reservoir through that reactivation approach is that even when cells are reactivated, they're often not cleared by the body. But there's good evidence that there is ongoing immune surveillance of the reservoir. We see that HIV-specific CD8 T cells are maintained even after decades of suppressive antiretrovirals. And proviruses that have elements that favor protein expression will decay faster over time. So this led us to be interested in the HIV accessory protein Nef. And one of the many things that Nef does is it downregulates MHC from the surface of infected cells. And MHC is the molecule that's responsible for presenting viral proteins for surveillance by CD8 T cells. So to test this hypothesis, we collaborated with Jimmy Dikiakos and his previous PhD student, Dr. Mitch Mimby. And Mitch cloned Nef sequences from 14 participants in our cohort and then used them to infect a T cell line that expresses MHC. And so then he could measure the ability of those participant Nef sequences to downregulate MHC by flow cytometry. And we then correlated the ability of a participant's Nef to downregulate MHC with that participant's individual rate of change of their QVOA data over time, so their slopes from the Bayesian model. And what we found in the bottom left-hand corner, you can see that people who had particularly poor MHC downregulation, so their Nef really didn't function very well, they had the fastest rates of decay. They had the most negative slope. Whereas people who had a really excellent functioning Nef, that's on the top right-hand side of the plot, those people either had no change in their reservoir size or even a significantly positive slope, implying a drift up over time. And so Mitch also measured the ability of Nef to downregulate CD4 from the surface of cells. This is another one of Nef's many functions, but it wouldn't be expected to influence CD8 surveillance at all. And he found no association between the rate of change of the reservoir and CD4 downregulation. So these data suggest that Nef might help protect latently infected cells by limiting antigen presentation and shielding them from cytotoxic T cell recognition. And there's also data from a cross-sectional cohort here in Canada. Sabrina Brumet's group has also found a similar observation. And I find this really especially interesting because unlike many host targets that we're targeting with cure modalities, Nef is a viral protein. So this raises the possibility of a druggable viral target that could be combined with latency reversal or immune-enhancing strategies. So if you're interested in the effect of Nef on the reservoir, Livia Kuckman will be talking about it this afternoon in the Basic Sciences Oral Abstract session, a different Nef function, SERINC5 downregulation. And then on Sunday morning, Maxine Nicolais will be talking about Nef inhibitors. So the final observation that I'd like to tell you about is the effect of an antiretroviral drug on reservoir dynamics. So before 2019, WHO guidelines were for first-line ART therapy was non-nucleoside reverse transcriptase inhibitor based. So one NNRTI on a backbone of 2 NRTIs. And then in 2019, the WHO recommended replacing the NNRTI with an integrase strand transfer inhibitor, dolutegravir. It has a much higher barrier to resistance and is much more favorable from a tolerability perspective. And what this means is that more than 90% of African people living with HIV who are on first-line therapy all switched to a dolutegravir-based regimen in a relatively short period of time between 2019 and 2020. So what that looked like in our cohort is we had people on efavirenz or nevirapine, and they all switched to dolutegravir, and the NNRTI backbone was all unified to TDF and 3TC. And this gave us a really unusually clean opportunity for an observational cohort to ask whether or not dolutegravir initiation is influencing the reservoir while still being able to control for things like efavirenz withdrawal or the previous NRTI backbone. So when we first So how we actually got interested in looking at dolutegravir was when we first looked at the QVOA data, we were really interested in calculating the rate of decay that's been reported in many North American, or few North American cohorts. And when we went to calculate the rate of decay, we found it's a flat line. There's no significant difference. And maybe this isn't so surprising. Most of the cohorts started our observation period when they'd already been on ART for 7 years, and we know that the decay is very slow at that time. But we were a little disappointed. We hoped we might see some decay. But when we looked at the data, we saw these spikes. So many people seemed to be coming down and then would suddenly have a spike in their QVOA outgrowth. And this was in both males and females, so it couldn't be fully explained by something like menopause. And when we went and talked to the clinicians in Rakai, we found out that these spikes were coinciding with this nationwide switch to dolutegravir. So Rusiel then realigned the data, instead of based on time since ART initiation, based on days since dolutegravir switch. And what we observed was a 1.7-fold increase in QVOA outgrowth right after dolutegravir initiation. And this was flanked on either side by decay, with a calculated half-life of about 7 years. But there's been many studies that have shown that dolutegravir has no effect on total HIV DNA. And so based on this led us to hypothesize that maybe what we're seeing here in this QVOA data is not actually a change in reservoir size, but is a change in inducibility. So to explore this further, my PhD student Sarah Gowanlock modeled this in vitro using 2 different cell lines, JLAT 10.6 cells and CLATs, and both of these will express green fluorescence protein when they're reactivated under the LTR, the HIV promoter. And so to simulate this, she pre-treated the cells with either dolutegravir or efavirenz, and then she stimulated them with TNF-alpha to recapitulate that QVOA. And what she found is that dolutegravir pretreatment, but not efavirenz pretreatment, significantly increased HIV reactivation in both cell lines. Sarah next wanted to investigate whether there was any immunological perturbance or changes in the participants in our cohort that were associated with this. So she measured a panel of different immune analytes in the plasma, and she measured them prior to initiating dolutegravir, within the first year after initiation, and then more than 1 year, so longer term. And what she found is in that initial period, within the first year of starting dolutegravir, there were these really substantial shifts in many immune markers. And we saw coordinated increases in cytokines that were associated with cytotoxicity. So things like interferon gamma, granzyme B, perforin, TNF-alpha. but that these changes were temporary, and most of them returned to baseline by more than 1 year post-dolutegravir, suggesting that this is some sort of a transient immune perturbation. And of course, we can't know, is this the effect of the reactivation and HIV being released from the cells, or is this the effect of dolutegravir itself, or probably both, but certainly something going on. So to explore this idea specifically of cytotoxicity, because we're really interested in the idea that immune cells, cytotoxic immune cells, might be recognizing cells that are getting woken up by dolutegravir, Natalie Howard and Andy Redd's lab at the NIH performed flow cytometry on PBMCs from the same time points. And she observed an increased percentage of CD8 T cells making interferon gamma, and also increased activation of NK cells in that first year after dolutegravir initiation with a return to baseline. So in summary, we observed a 1.7-fold increase in QVOA outgrowth with a regimen switch to dolutegravir. But again, this leaves us with the question, much like the male-female data, is this QVOA, this change in QVOA outgrowth, due to intact reservoir size changing, or is it just due to inducibility changing? And in support of the latter, in vitro, we observed that dolutegravir is able to increase reservoir reactivation. And we also saw many transient increases in markers of cytotoxic activity, suggesting increased recognition of HIV proteins. But what we're left with, and what we still don't know, is whether this perturbation in the reservoir is sufficient to reduce intact reservoir size. Or is there going to be viral immune evasion from things like Nef, or clonal expansion from the immune activation, so that there's no net change in the overall reservoir size. And of course, we'll need assays like the intact proviral DNA assay to be adapted to global cohorts to be able to do this, which we're very, very close to being able to do. So I'm very excited to have Isaac Wu's talk this afternoon about how in-stage therapy is shaping integration site profiles, because I think it's possible that things like clonal expansion or deletion of lineages through cytotoxic T cell recognition could be going on in the post-dolutegravir period. And then, of course, we're left with a really big question of how. Like, how is dolutegravir making cells easier to reactivate, which I think is a really important question from moving forwards in terms of cure development. So take-home messages that I would like to leave you with after this talk is that longitudinal, globally representative cohorts can be very informative for understanding real-world HIV persistence. And we've observed that host biology, viral factors, and even regimen switches can all shape the reservoir. I also really want to leave you with a message that inducibility and reservoir size are not the same thing, and that they're likely both very important attributes of the reservoir when we're thinking about curing people of HIV. And finally, I want to leave you with what I hope is a very hopeful message, in that reservoir inducibility is dynamic, and the reservoir can be perturbed outside of just the lab or a very controlled clinical trial setting. And so the last piece of data that I want to leave you with is this graph. And so what this graph is Russil's model of one person over time of their QVOA data. And if you just look at this graph, you might think that their reservoir is gradually increasing over time. But if you actually overlay the exposures of menopause and dolutegravir, what you can see is that this person actually just experienced two blips, two increases in their QVOA outgrowth, and that this was flanked with periods of decay, which I think is hopeful. So I want to end by acknowledging the many people who have worked on this cohort and whose data I presented today, especially very much the team at the Rakai Health Sciences program led by Ronald Galiwango. Tadeo and Paul do all of our PBMC isolation. Javita and Bridget are the ones in the labs doing the IPDA assays. Anthony and Agri are our data managers. Edward Kankaka is a junior PI. Sarah and Margaret are the lab managers, and Stephen is our clinical officer. I'd also really like to acknowledge Art and Russil, who developed that Bayesian model that have allowed us to really pull such confident observations out of the cohort, and Jimmy's lab for the assistance with the Nef work. At REACH, Gwyn Lee is the one who did all of the full genome sequencing. Mark, thank you very much for the CLATs, and Sabrina Brumet is working with us on the IPDAs. And then, of course, finally, I want to thank the National Institutes of Health, who supported this cohort for 10 years, and Andy Red, who I worked very closely on this work. And the last thing, the most important thing, is thanking the participants, because without their extraordinary dedication to this study, we have less than 10% follow-up over the decade. None of these observations, of course, would be possible, and I really hope that we're going to be able to bring them an intervention trial in the very near future. So thank you very much for your attention.
Is on now? Oh, excellent. Thank you. Well, thank you so much for that excellent talk, Jessica. We have plenty of time for discussion. I presume there are some microphones circulating. AV team. There we go. Or we have a volunteer with a microphone, so just feel free to flag them down or over there as well. Yeah. Jerome, you're up. You have a microphone right behind you.
Thank you. Thank you so much. I'm wondering whether you assess the subset of T cell, if there is some association between the fact that you are capable to reactivate better in some population, maybe in which we have some difference in terms of proportion of central memory or effector memory cell, that may explain in some case some difference between DNA reservoir versus the capacity of a cell to be restimulated. That may explain maybe some difference that you observe.
Yeah, I definitely think so. So the question is, I've been requested to repeat the question. So the question, I believe, is that is it differences in the T cell phenotype that's dictating how easily a cell is reactivated or not? And we haven't yet looked at that. So we haven't done like cell sorting and then looked at reactivation in the different T cell subsets yet. But what we are doing is Natalie Howard is doing a really in-depth T cell phenotype of memory cell subsets before and after dolutegravir, so we might be able to see if those T cell subsets increase.
Yeah.
But cell sorting is definitely something that we would like to do in the future.
Yeah, because during the recovery, probably we change a little bit the proportion between central and effector memory cell that may explain maybe some part of the increased capacity to reactivate a cell.
Yeah, I definitely agree with that hypothesis.
This side of the room.
Hi, Jessica. Daryl Tan from Toronto. Fantastic plenary. Thank you so much for that. I was interested in the observations you made around dolutegravir, which sound like they were pretty opportunistic, just because programmatically the whole country changed over, as so many other countries have done. And of course, I'm curious, in our context where we use DOL, but we also use other integrase inhibitors, and there's newer drugs coming down the pipeline, what else do we know from your lab, from other labs around the impact of other commonly used drugs in our context, bictegravir, for example, on these same assays?
Yeah, so I think the data is really new, and in some ways is very limited, because it is so much easier to measure total HIV DNA than it is to do any of these intact proviral assays. And that signal, because 95% of the virus is defective, and it's not, reactivating and being cleared in the same way. So it really inhibits your ability to measure a signal specifically in the intact compartment. And so a lot of the information that we have right now is just from total HIV DNA. And of course, in our cohort, we only have all of the drugs were provided by PEPFAR. Please, PEPFAR, don't go away. So we don't know about anything other than dolutegravir. But Sarah is going to be testing, first of all, we're looking at the mechanism. So I think if we could get an idea of the mechanism of what's happening, and we think dolutegravir and other integrase strand inhibitors chelate cations, and so we think maybe it has something to do with calcium influx. I think if we could get at the mechanism, we could have an idea if other integrase strand inhibitors or other ARTs were going to have this effect. And then, of course, using the cell line models, we'll be able to test those other insties, in a very controlled fashion. And so I hope that within the next year, maybe next year by CAHR, we'll have some really interesting data to present on that. I definitely don't think, I don't know, and I don't have any data to support this, but based on the data that I do have, I don't think this is at all like a safety concern for dolutegravir. In fact, I think it may actually be exciting. Like, this could potentially be a moment where maybe we can reduce reservoir size or learn from it to reduce it. But I mean, that sort of excitement, like, this isn't the first thing that we found that's been able to really reactivate the reservoir and then has had no effect on total intact size. But definitely, I'm not thinking that bump in QVOA that we see is actually an increase in the size of the reservoir.
Over here.
Thanks very much. Chris Powers from the University of Alberta. I'm wondering about the effects of dolutegravir on other sort of deep tissue reservoirs, like myeloid cells. Do you have any data on that or any thoughts?
No, but we are going to start sorting before IPDA. So we're discarding the monocyte fraction right now in our QVOA protocol. In the IPDA, we're doing total CD4, but we're going to try to start splitting. And of course, the limitation of that is cell numbers. Like, these assays take so many cells, and we don't want to be doing enormous blood draws on people. But yes, we're going to get that, and I have no idea yet. I'm really interested also.
What about sex effects on that reservoir?
So for dolutegravir, we controlled for sex in the analysis, and we saw the same effect in both males and females. And we also controlled for like efavirenz withdrawal, NRTI backbone, all those sorts of things, and we didn't see any significant differences.
So we probably have time for 2 more questions, so we'll go over here, and then Nick, I'll give you the last word.
Hello, Santiago from Queen's University. To your left. There you go.
Yeah.
Oh, thank you. And
You're
I'm like totally blinded by
You're in the fireball.
lights, too.
It's okay, very quickly. Thank you for this excellent presentation. We know distribution in tissues is different, the reservoir distributes differently, and I was wondering if all our studies in QVOA, it's usually using PBMCs. I was wondering if there's differences if we try to reactivate virus from different tissues.
Yes, I think that is an excellent question. And, a lot of HIV research and answering questions like that are done in really small populations in North America and Europe, where you can, more easily go in and do lymph node biopsies and things like that. Not that we don't have that capacity in Uganda, but we're in a pretty rural area in Rakai. Not as well suited to like sort of a long observational cohort, right? Different studies have different strengths, which is why we need them all. And I totally agree that's a really interesting question. Probably not one we can answer with our cohort unless we just took a limited subset, but definitely so important. I completely agree.
Nick.
Hi, Jessica, over here. Nicolas from the University of Montreal. Beautiful talk, really beautiful plenary, and I'm very much impressed by the number of VOA assays you were able to run. I know this assay is just a pen, so congratulations on this. On the dolutegravir story, I think an alternative explanation is the fact that this efavirenz has been reported as a TAC molecule by Merck, actually, and Merck is now developing other TAC molecules, which are molecules that are basically killing cells upon reactivation. So the lower amount of IUPM you're measuring in people on efavirenz is maybe because in the assay, during ex vivo reactivation, you're actually killing these cells, which you don't anymore when people are on dolutegravir. You see what I mean? So I was wondering whether this is something you can explore, because if that's the case, that means that actually efavirenz is better than dolutegravir, right? Well, better, I should not say that, but would kill reservoir cells.
No, like we had exactly the same thought, and in fact we talked, the TAC team from Merck asked us to look in our data, and so what we did is we looked in the cohort pre-dolutegravir, because we had half of the people on nevirapine and half of them are on efavirenz, and we were really excited. We thought maybe we would see a difference in the reservoir in the people who are on efavirenz, and we saw no significant difference. And we worked with Daniel Rosenblum and looked at that data from all different angles, and we didn't see any effect. So, I mean, I think because we have, we weren't going from a unified NRTI-based therapy to unified INSTI-based. We had the ability, and with 90 people in longitudinal sampling, we had that ability to look for an previous ART treatment. And I think, and especially with Sarah's in vitro data, we really do think it's from dolutegravir initiation. I was hoping we'd also see efavirenz, of course, having that effect. Maybe they could be combined, but we didn't see it in our cohort in the pre-dolutegravir era.
So unfortunately, we're at time, so we'll have to leave it there. Please find Dr. Prodger afterwards to discuss that amazing data. Thank you so much.